comparison. And the F1 propanol, you know, there's, again, no difference. It's around 0. So you have, between both the propanol M71 and the home M71, in response to the acetophenone, there's not much of a startle compared to the ones that have been preconditioned in the F0. So the offspring are having a greater sensitivity. So what they then do in D is they look at the effect to propanol. I'm not sure why they didn't do that with the C57, but they do it with the M71 mice. So the parents have been conditioned to propanol. Yeah, they have a greater response, and the acetophenone ones, you know, doing the statistics on it, it doesn't seem to be, you know, there's a big spread there, but you know, it's closer to the home-treated M71 mice. So all of this basically lays out the groundwork that the mice have been sensitized. They have a greater startle response in the F1 generation to this odorant when, you know, in response to the parents being treated this way. So any questions about figure 1?
I have a quick question. I may have missed this, but did they do like repeat exposures, like the same animal gets a repeat exposure, or is it just like the N of 16 different animals, a one-time deal?
Yeah, that's a great question. They mentioned something like that in the paper. I'm blanking where it was. They do make some sort of statement regarding not having to. Somewhere it said about, you know, obviously the problems with repeating the mice because then you're conditioning the mice while you're doing the experiment.
I see. Yeah.
There is some sort of I'll have to find it later, but I think they may have talked about that.
Okay, I'll look for it. I'm sure I just missed it. Yeah.
Yeah. So what they do in figure 2, we're just going to move on to that one, is they have a different assay. Again, it's increased sensitivity. So these are, you know, so right now we're setting the groundwork for increased sensitivity in the F1 mice. And it's a different assay. It's basically not a startle, but a box. And they have 3 different chambers. And one of the chambers has the odor, and the other one doesn't have anything, and the other has just an empty container. And it's called an aversion index. So I think what happens is the mice walk around, and wherever they count how long such a mouse lingers in a certain area, and the less it lingers, that's greater aversion because it either doesn't like the smell or it's sensitized to it. So what they show here, they have different acetophenone concentrations, and when they have the concentration really low, 0.01%, this is figure 2A, they're comparing acetophenone conditioned C57 parents, F0, and F1 from propanol conditioned parents. If they look at acetophenone, you see a really, you know, according to this, a dramatic difference in the mid-range. So 0.03%, the acetophenone treated parental strains gave birth to offspring that are really don't like the smell. And by don't like, what I'm going to emphasize that it's not a fear response. It could also be that the mice are just more sensitive, which is borne out by the fact that they have more receptors. So they have an aversion. Interestingly, yeah, the inversion doesn't happen at a lower concentration, and the aversion kicks in also for the propanol treated mice at a higher concentration. So these mice are really, you know, this suggests to me that they're really more sensitive to this odor, which the later experiments bear out because they have more of these receptors, more of these neurons to pick up this odor. And they do the same thing with propanol. So they show that the propanol, you can train the mice, you know, not train the mice, but the same phenotype can be propagated to the next generation. So they look at 0.003%, and this F1, what is this? Yeah, so interestingly, the F1 acetophenone treated mice, I guess the error bars, you know, it's around 0. They don't have an effect, but the propanol treated parental strains, F0, this phenomena passed on. So it's not just acetophenone, but it's also propanol. So these experiments kind of lay the groundwork for what comes later. And I just want to also reiterate, I'm not sure if it seems to me, I've been looking to see if they did this control here, but it seems to me I'm not convinced that it's an actual connection to a fear response and the smell acetophenone, because I don't know if they did an experiment with the F0 where they just subjected the male mice to the odorant without the condition. In other words, is the electroshock, is the conditioning even necessary to have this increased sensitivity? Based on earlier work, which suggested that, you know, having acetophenone in a diet of the mice will cause an increased smell response. So is it?
Oliver, I have a question.
Yeah.
Do we know, or do they mention, like, what is the normal response to this chemical or whatever, you know, for a normal mouse? Do they like it? Do they go away from it without any behavioral training? What's the normal?
Yeah, well, I think that's in figure 2, right? So if the concentration goes really high, there's an aversion to it, right? Which is the F1 the F1 ACE and F1 propanol. So the F1 propanol is a control. So
yeah, I guess what's unclear to me, which I think sounds like the same thing that's unclear to you, is like, it just, does more receptors of anything necessarily mean more aversion to it? Like, it seems like that's something that's tacitly assumed here.
Well,
but
I think, and again, I haven't read any of the background. There's a lot of background information on this, especially dealing with acetophenone and receptors. But I would think for any kind of animal that has a very, very high sensitivity, that high levels of anything is going to be an aversion response, you know, like too much of a cologne.
What about if it's a food or something like that? Wouldn't you, you know?
Right, but I think
it can go either way, is what I'm saying. Something that you really want, but you want to go away from.
If you look at a figure 2, you actually will have your answer, because when the concentration of acetaminophen is 0.3.
No, yeah, I see they're averse to it. I get that, but
No,
but also there's a lot of stuff going on in here, so I just want to.
Listen, if the concentration of acetaminophen goes to 0.6, even
everyone's averse to it. I get it.
Both averse, and the same happens for propanol. That's your answer. You know,
I know.
They just averse to that smell. You make it powerful enough. It doesn't matter if they were prepared.
Yeah. I'm asking a more fundamental question about just, have they explained what exactly the mechanism here? Why, you know, what physiological things does it do to them? Why don't they like it?
I don't know. That's probably happening at a higher order brain function. It's probably overriding some kind of circuitry that's just saying that, you know, there's too much of a, you know, either you condition an animal to a smell where it doesn't smell it anymore, or it's so potent that it basically. I
mean, you can correlate it with yourself. Is there any smell that you can stand when it goes to extreme?
I guess if there's too much, I like cheese, but too much of a cheese smell would
probably
knock me out.
So.
Dave, what are you smelling? Almonds. Okay.
Yeah. So this is supposed to be like a cherry almond smell. The other question I had was, this may have been addressed in an earlier paper, but all of these responses, I'd like to see how. It'd be interesting to find out multi-generationally, how similar this mouse's responses are to the F0, right? Because the F0 is conditioned to this, and we're getting this response, but are the parents more sensitive, or is this going down generation-wise? I would think that this would be kind of a neat thing to ask is this effect being diluted from F1 to F2 to F3 compared to F0, because they don't show the F0 data here for how responsive this mouse is.
Yeah.
What I think would be an interesting experiment to follow this up, if assuming, like Dennis is saying, that this is just a chemical that they're naturally averse to, would be to take a chemical that they're naturally positive towards.
Well, no, I don't think they're naturally averse to this chemical. I mean,
because if you look at the graphic, the F1 prop mice, they averse if the concentration goes to 0.6.
Right, but at a low concentration, none of these mice are averse.
Anyway, what I'm saying is I think it would be really interesting to take something that they're really naturally positive towards and turn that into a negative through this, basically, fear conditioning, and then see the difference between the F0 and F2, because then that would be a total reversal of the F0 condition, where in this case, if the natural response of the unconditioned F0 mice is aversion, then you're just looking at like aversion and a slightly greater aversion. It's easier to get confused, where if it's positive versus, if F2 is really averse and F0 is really, drawn to it, then that's a huge, unmistakable difference there,
The other thing that might tease out is right now it could be that they're just more sensitive to the odor, but they haven't really inherited a behavior associated with that. If you could alter the behavior and the sensitivity, that'd be a lot more enlightening, I think.
Yeah, that's what I'm saying. And then it's much more like what you're looking for, Oliver, like actual fear, or something is more of a behavioral thing and not just an increased sensitivity to something they already don't like.
I mean, the question is, there is probably one thing that we don't know about mice psychology. It's probably for mice, it's naturally whatever they sense, they're just afraid of everything. If you can make that assumption, then the assumption they do in paper is valid.
Well, we'd have to go to the literature. Maybe there is something that even high levels there. And I see what you're saying, Keith, is switching the, switching the effect would be one thing, but. And that kind of gets, I think, to the heart of where I had a problem with this paper, is that the assay that they do, this odor potentiated startle, is sort of a, I don't know how you can say that it's you're transmitting a fear conditioned response rather than just increased sensitivity, because you have more neurons being produced in response to this odorant. That in of itself, I think, is an interesting phenomena. You're having a trend, at the very least, you are having in the F1 and F2 mice, you're getting an increased sensitivity being transmitted multi-generationally. But I don't really, unless you do an control where the parental mouse was subjected to the odorant without a conditioned fear response, or you do something like you suggested, Keith, which is have a positive, response turned into a negative, where you now have this, correlation. But you would have to do then an analysis later of some kind of a fear or some sort of more a finer grained analysis, where it's not just a generic startle response.
How you distinguish fear from aversion in mice?
I don't know.
You talk to them?
I don't know. You can talk to some animal psychologists. Maybe there's some way.
But you definitely could distinguish between aversion and wanting something.
Yeah.
So that's going to be easier to see.
Yeah, but still, it will be aversion. My more general question is how you distinguish fear from aversion. The whole idea that you can distinguish in mice fear from aversion looks funny to me.
Well, yeah, but I'm saying that the problems of this paper might involve the difficulty of that distinction, where if you have the other situation between, being drawn towards versus aversion, then you don't even really need to worry about that nuance.
I think they unconditionally proved that you can transmit epigenetically the aversion feeling, so you don't need an extra experiment to check it.
Well, it's what they've proven here. If they've proven it, is they can increase sensitivity.
Yeah, and aversion. No, aversion.
Not necessarily aversion. It's that
So here's the definitional problem we have here.
Yeah.
Increased sensitivity seems to correlate directly with aversion.
Yeah.
So you can't really separate the two. You can't say that they're sensitive but not averse. It seems like if you're sensitive, they will be averse because they're
right. Yeah. So it seems like the baseline condition is at least some level of aversion. So by increasing the sensitivity, you're increasing the aversion.
Right.
No, I think the paper is great. It's just they probably went for more pop culture name instead of using mice transmit vigilance. They say fear. I think it sounds better.
Well, but that goes kind of to the root of it, because I mean, the heart of the whole paper is a conditioning of a fear response, and really that's half of the claim, is that not only the increased sensitivity is transmitted, but the fear is also transmitted, which they don't really measure in these follow-up generations.
I mean, if you look at this from evolutionary perspective, if you can inherit version or sensitivity, it can equalize to to better survival. Because you will learn really fast to fear it, because you will sense it better. You will see the predator from far away. So from evolutionary perspective, it doesn't really matter.
Yeah. Well, what do you mean it doesn't really matter?
It doesn't matter because if you're more sensitive to smell to an animal, to a predator, you'll pick it up from far away, and you will very fast learn that. Oh, I felt it.
Yeah, I want to unpack the implications of this later. I want to pursue the next.
But Oliver, it seems like it's borne out by the paper. Like they explicitly mentioned that they don't do that in this paper. They don't really assess, the psychology of it. So they're cognizant of that weakness.
I know. Well, they are, but then when you read there's a News and Views article that writes about the paper, and the third big thing that's written about this paper in the News and Views article is that I'm just quoting from the News and Views article. Third, not just the memory for the odor, but also the association of the odor with a fearful experience is transmitted across generations as a conclusion of this paper. So.
Maybe that's just a little bit of sensationalism, or?
Well, anyway, that's the conclusion that seems to be being reached from the data. But all right, so going back to the sensitivity, if we look at Figure 3, that's pretty clear. I mean, what they do is they look at these M71 mice that are expressing beta-galactosidase, and they're looking at these regions where these neurons are, that are specific to this odorant. You don't really need to do statistics. You can see that F1 acetophenone treated, offspring from F0 mice have a lot of these, neurons that are specific. So the analysis is kind of icing on the cake. There's clearly more stuff happening neuronally in these mice that have come from parents. So yes, you do have more of these receptors, more of these neuronal connections, and more of this sensitivity. So I think that's, a pretty good, that in of itself, that data is pretty good. Figure 4. Figure 4 basically takes it a step further. So we've been talking about F1 mice, and they want to take it to the next generation to see if this really persists. So they've looked at F2 mice, and they've also looked at these in vitro fertilized derived generations, these IVF mice. And I think they're looking at F1 IVF. And the reason why is just to further put a barrier between any kind of nurture effect versus nature. So they really go out here pretty far, and the effect persists, which is good. So again, they do these same. I'm not going to go through these step by step, but they do these the same analyses that they did in the last two or three figures. They look at they do the staining in figures C, D, and E, and F. They look at the odor potentiated startle. It still persists. I'm looking at the numbers here. I don't know how they designed these experiments, if you could really go across, comparing Figure 1 and seeing whether or not the effect diminishes. That might be a little hard because it is kind of multi-generational and you're having new experiments. I mean, it would be kind of cool to see that effect, and see its persistence across the generations and how fast it diminishes, and kind of get a number behind that. That would be really, really cool. But of course, that would be a long-term experiment, to say the least. At least with mice. Maybe you could do it with another model organism, because they did in a lot of these in the references they mention these epigenetic phenomena certainly happened in I think locusts, insects, and of course they happen in single cell organisms like yeast. So you can use that as a model organism. So that's all I want to say about Figure 4 is that this is a persistent effect that goes on multi-generationally. And they basically do, I don't want to spend too much on Figure 5. Figure 5, it's the next one. Thanks. Is they do the same analysis, but now they're looking at, they do this cross-fostering. So just in case the mother in which it was weaned had some kind of influence, they take the pups and they give it to a foster parent. foster mouse, and they see if this effect persists, and it does. So it's not due to fostering. So again, this is just, more demonstration that this is clearly a inherited response that is most likely not due to any kind of influences from the parents outside of the fact that they produced the germline and propagated these mice. And that what you're getting here is more of these neuro and anatomical differences that are due to this treatment with the acetophenone and the F0 generation, and that you get this increased sensitivity through this, this OPS, this odor potentiated startle response. So in the supplemental, they actually do these experiments with, so I'm not going to go over the supplemental, but in the supplemental, if you read it, they do these experiments also with the maternal line. So I think they repeat, maybe not all of them, but certainly some of them they repeat with the maternal line, and you get the same response. The interesting thing about this fear response is that they do an assay, and this is again in supplemental. The mice are not generally more stressed out, so when they do these fear conditioning responses, they try auditory fear conditioning, and they basically are not more, they're not more prone to being fear conditioned, which I think is interesting, and which to me suggests that, again, that this might be solely due to increased sensitivity, and it's not a fear response that's coming along for the ride as a condition. Again, I mean, Keith, you suggested one type of, experiment that might provide another control. I would think just having a control with an odor without any kind of conditioning, and seeing whether or not you repeat the same type of sensitivity without the need for any kind of electroshock.
Right, because they note that there was previous papers where there's some sort of in utero effect, right?
Yeah.
If the parents were exposed more, then they would become more sensitive. So yeah, it stands to reason that would maybe have the same effect as the process.
Yeah, so I think what's solid here, with the major advance to me for this paper, and there's one more figure 6, which we'll go over, but so far this major advance is that you're getting this increased acetophenone sensitivity and also propanol chemical sensitivity, and it's persisting all the way up to the F2 generation, and it's absolutely nothing to do with any kind of nurture effect. Any which way they mess around with the mice, foster parents or IVF, it still persists. Whether or not it's really two memories, I think that's still an open question, right? Whether it's increased sensitivity coupled to fear, I don't think that is addressed really here. So they at the end, they really want to tackle a biochemical question, which is, well, what's happening here? How is this effect persisting? And one kind of one good guess is that it's an epigenetic phenomena, since this this phenomena has been, and I sent out a review paper that looks at epigenetic phenomena that are transmitted generationally in mammalian systems. This is already known in mice and humans, actually, for dietary restriction, where you have children and grandchildren who have increased incidence of diabetes due to their parents being under starvation conditions. So this is a good place to start. And where they start to look at is they look at the sperm cells, the male mice that have been treated, and also the F1. I don't know if they look at F2. They have here F1 mice, and they want to look at this methylation status, the CPG sites in the OLFR 151 location, the locus. And they show you the sequence later where they look. I can't remember exactly where around the gene they looked. So they do this bisulfite sequencing, and they basically see a difference that's statistically different according to their scale here, percentage methylation. And this is I think one general area that they look at, and this is figure A. So they look at propanol treated sperm, F0 from parental strains that have been mice that have been treated with propanol, F0 from acetophenone sperm, and you see this decreased methylation. See, propanol, overall CPGs. And they look at another receptor site, OLFR6, which has nothing to do with this, and there's no difference between the propanol and the acetophenone. That's figure C. So they're looking at different sites. So B, D, and F are basically looking at different regions, different CPG islands, CPG 1, 2, 3, 4, 5, 6, 7, 8, 9. So not all of the islands, not all of the regions are, differentially hypomethylated, only some of them, and I think only one of them, CPG2. The interesting thing is there is, this I think experiment raises a lot more questions than answers. There is some kind of methylation, a demethylation happening, but it seems to be only correlated with the sperm locus. It doesn't, when they actually look at the cells from the glomerulus, the somatic cells, I don't know if they have data here, but that methylation is not that demethylation is not present. So it's only in the sperm. It could very well be that this effect, and this is just speculation, happens. This demethylation is only required during early embryogenesis, and then is somehow reset in the somatic cells. But then it has to persist in the germline precursor cells, obviously in the F1 and the F2, for this phenomena to be propagated. So it seems a bit odd that it's only happening in the sperm and it's being somehow reset in the somatic cells and then maintained only in the sperm. That seems to be what they got out of this. In the supplemental, they look at several different loci. So they looked at other modifications and they didn't see a difference. So they looked at acetylated histone H3. They looked at trimethylated histone H3. They looked at a couple of other different epigenetic modifications, and they didn't see a difference. So it was only this bisulfite sequencing reaction in this one CPG2 island that is only in the sperm cells of the mice where this phenomena is taking place. So that's basically where they kind of like leave this at. One thing they mention about the spermatogenesis is that they when they did this F0 conditioning, let's see, this is at the end of the paper. Interval of 13 days. Yeah, so they mention at the end that they're, this is, in a sense, it's a good first start, but more needs to be known. They talk about, what fraction of these, obviously the mature sperm have this hypomethylation here. What about sperm precursors? What percentage? Mentioned they don't know the fraction, so maybe if you could do some sort of fluorescence activated cell sorting to determine this. So clearly a lot more has to be done. So that's basically the paper. There are clearly big implications here if there is a connection between a learned memory, such as fear. One implication that they don't mention here, but I think actually Conrad Waddington mentions going way back in the 1940s and 50s, the implication of the phenotype to basically influence the genotype later. So I worked with yeast in grad school, and one thing that we do know is that epigenetic changes can also influence the mutation status of DNA, right? So if you have an area that's silenced, basically, the chromatin is deacetylated, it's in a more compact configuration. Not only is the transcription levels go down, as you see in this case, but you also have the DNA more prone, at least in certain regions. So certainly in ribosomal DNA locus in yeast, which is very prone to mutations, you get a much higher frequency of mutagenic events. In this case, it was that I was looking at specifically recombination events. but essentially the DNA is more fragile. So in a sense, you could posit that, and I think Waddington may have proposed this as a way for more permanent changes to be affected at a locus by having essentially the epigenetic status altered. And this may be a mechanism that can take place. And I don't know if I'm not sure if experiments were, how many experiments have been done to kind of probe this, but this could be an interesting phenomena to look at multi-generationally to see if certain phenotypes could be locked in a more permanent Darwinian sense by having this overlay of Lamarckian epigenetics kind of, influencing the frequency, basically funneling the mutation frequency into certain loci. That's something I don't think they mentioned in this paper, but that's something that's, I think, interesting from an evolutionary perspective. So I think I'm going to end it at that with this paper.
Say, Oliver, have you come across any of the research about epigenetic memory in C. elegans?
No, I honestly, I haven't been looking, so I don't know. So were there any, I wasn't even aware that C. elegans was being, I guess, I do know that C. elegans, and you probably know yourself as well, probably more than I do, that they, C. elegans, has neurons that respond to food and they have a lot of genetic mutants that can respond to different food sources. I think E. coli is their favorite food source, different chemical cues. So I don't know if they've done epigenetic analysis with C. elegans to make these connections, but I think that would be a great model organism, certainly better than mouse, because you can really crank through the generations here.
I was just asking because I know they've done studies about longevity, inheritance, and also the ability of C. elegans to learn mazes, where they basically would teach one generation an ability to learn maze, ability to navigate a maze, and then they'd actually grind them up and feed them to a subsequent generation. But that actually suggests a different mechanism of inheritance than.
I remember now that you mentioned that, that's very familiar. Yeah, ground up worm and then the memory being transmitted by eating another worm. It's kind of reminds me of the reason why cannibals eat their victims, right? To achieve the power of their conquered enemy. So you achieve the power of the conquered worms. Now you can memorize the maze. But one thing,
ancient blood drinking rituals also, where you would drink not just enemies, but patriarchs and of the society.
Yes. So maybe it works. But going back to the worm, they're in all the so it's the way. And I don't work with worms, but one way that they basically silence genes in worms, I think, is that they have silencing RNAs. So these basically microRNAs and these small RNAs that are complementary to the RNA you want to knock down, expressed in E. coli that the worms then eat, right? So then that would influence the expression of genes. In one of the review papers, so RNA is known to be an epigenetic factor. Certainly these you have RNAs that like these how's it pronounced? Exist. XIST. So you have RNAs that code regions of the genome that can basically that can shut down regions of the genome. So RNA does play a role in epigenetic inheritance. So grinding up a worm and feeding it to another worm and getting some sort of phenomena that then is propagated. It's certainly that's the technique they use with E. coli when they feed it, and they express RNAs. So that certainly could be a mechanism by which you can get epigenetic transmittance of information that can then persist multi-generationally. If those RNAs, I guess, linger and are still stuck. If you have enough of them, I guess. I don't know.
I have a question, Oliver. I think this is related to what you were saying before about how it would have been nice to have compared the level of the response from F0 to F2. Basically, my question is, how is this reversible? Because you'd think like it's not evolutionarily speaking like ideal, at least for this, one family to, once you've been, overstimulated by this one thing forever, your whole, descendants will always be like oversensitive to this. Like, does it gradually fade away? Is there some sort of mechanism that could undo this demethylization? Is it reversible, I guess is my question. And as a follow-up to that, if you wanted to really kind of seal the deal on this kind of experiment, I'm not sure if this is feasible with the technology, but after this conditioning has happened, can you then take a sperm and then sort of restore its methylization status back to what it normally is, and then show that the offspring do not exhibit the response? So that would be another way to really solidify that it is that's the mechanism.
Right. Yeah, no, that would be pretty clear cut if you were able to in vitro change the methylation status in that locus in the sperm, and then do the IVF and then show that the response went away. I think then that would be a really strong correlation. And as far as persistence, I don't know. I think there's tons of great experiments that still need to be done. And to me, this suggests that it's like other epigenetic phenomena, they can be reset. You have, methylases, demethylases. Methylation status is much tighter than, transcription status by being regulated by activators, but it is certainly much more reversible than a mutation.
Is there sort of like a future therapy here for inherited post-traumatic stress disorder or something like that?
I mean, yeah, there's a paper, there's a book that was just written where they talk about all of these. I'm blanking on the title, but it basically was an introduction to epigenetics, and certainly post-traumatic stress syndrome is being now, people are proposing it as a potential candidate for epigenetic modifications that can then might be reversible. There's certainly cancers out there that are now known that
there's a news today in science about the potential medicine, epigenetically based medicine. That's what they say.
Epigenetically, based medicine, exactly. So there's cancer. Oh, so there's somewhat up to date, not much, but yeah, certainly there are certain tumors out there that aren't the result of permanent mutations in the genome, meaning base pair changes, which is sort of the classical, definition of a cell that's become tumorigenic, but rather epigenetic modifications that will then turn on a certain gene that's perhaps a proto-oncogene or turn off a tumor suppressor. But this is more reversible. So if you can then, getting at a mutation that's locked in, having a base pair substitution or a big chunk of DNA missing is really difficult. Gene therapy is difficult, but swallowing a soluble molecule that can basically inhibit, a methyltransferase or a histone acetylase or a deacetylase is much easier. So, people are already, I think there might even be drugs, if not on the market, certainly experimental, that are being already employed in clinical trials that influence epigenetic modifications as cancer therapies, and that have been somewhat successful. I don't know if they're really broadly.
Is that
this paper in Nature is a follow-up for this study? This paper, they treated these mice epigenetically modified with a drug that reversed the methylation and the sensitivity goes down.
Oh, really?
Its drugs helps to dull bad memories. It's in Nature.
So they give it to the. Well, I guess we'll have to read that at some point.
Yeah, drug can improve the effectiveness. Blah, blah, blah.
so that?
Yes, I will send a link.
That presumably that has to be targeted at the specific gene, right? Because there's other places where the methylation's a good thing.
Yeah. That's what I know. Okay. So there's lots of these enzymes that do these functions, and but they hit a lot of different locations. So I don't think there's like one particular methyltransferase for one region only. So I guess I also have that question: is how you know, you're taking something that basically resets methylation or acetylation status or any of these other modifications, how specific is it going to be just to that locus? So you can show that it reverses the effect of that locus, but it's probably reversing the methylation status and other epigenetic statuses elsewhere.
I mean, could you modify, I mean, I don't know much about methyltransferases, but could you like add an additional specific DNA binding domain to?
Yeah, you could. Actually, there was a paper published a couple of years ago from a lab, Carmela Haynes, who used to be at the Silver Lab. She published a paper, I think it was in 2011, where they basically made targeted methyltransferases or histone deacetylases. I can't remember one or the other. Basically, they tethered like an activation domain, and they had those enzymes. I think it was components of this complex. It's got that complex name. It was found in the polycomb complex that's used in chromatin remodeling, and they targeted it to specific loci. I mean, the problem with that is, right, that it's a protein complex, so it's not like you're targeting a specific drug molecule to one particular one specific region and one specific protein that affects the epigenetic status. It basically you know, they made basically a fusion protein that only alters the that only alters the epigenetic status at one specific location. Mm-hmm. So I think that may have been actually the first paper where they did this, because I think they made a big deal in that paper where they said this was the first synthetic biology approach to making targeted polycomb factors or targeted epigenetic modification enzymes that can be tailored. So I don't know of any other paper since then that has done this.
So they use this histone deacetylase inhibitor.
Okay.
So it's just
there's a lot of them, yeah.
Yeah. So that's the thing they use. I know.
Yeah, there's a lot of HDAC inhibitors, histone deacetylase inhibitors, and so because there's a lot of different histone deacetylases, like the sirtuin enzymes are histone deacetylases. So it would really depend on which one they used, or if they used a cocktail. Yeah. Yeah, I'll have to look that up. Everybody's still up, right?
Yeah. Oh, that was long silence. The other thing I wanted to bring up, and I know Oliver's the resident expert on this, is well, I was going to talk about telomeres, which I'm not.
Oh yeah, I worked on telomeres, so a little bit, yeah.
It's also been shown that children of abuse, or those that you brought up PTSD before, potentially have shortened telomeres.
Yeah.
And I had a question about that, because I mean, I really don't know anything about it, but I know there's like this set point that you inherit, mm-hmm. that determines the length of your telomere. Mm-hmm. But if you, and then some people are saying, so if like you are an older father giving birth to, or having a child, then obviously the father's not giving birth yet. That will happen. But if you're an older father and you have a child, then it's potential, like the potential to have longer telomeres, that child having longer telomeres is also possible. Like, so I don't know, what is that there? Like, what's inherited and what's not inherited? And if someone's a child of abuse, or like, is the set point that is inherited different than if the individual was not abused? And,
yeah, I mean, I don't know. I did, there were some, I didn't read the actual papers where they correlate, supposedly the stress response and depression with like telomere length. I mean, and there's also the whole controversy is what is the actual role of telomeres in aging per se, right? So telomeres are clearly critical for the population, maintaining a population of cells. You don't have, so just briefly, telomeres are just the ends of chromosomes, right? Chromosomes are linear in most cells except for bacteria, and every time they cell divides, the ends shrink. At least one end shrinks because of the lagging strand, replication. You need a primer to synthesize the ends. So the enzyme telomerase just basically carries along its own little primer, and basically it's a reverse transcriptase and helps re-extend the ends so they don't keep shrinking. And supposedly, if they shrink too far, it'll look like a double-stranded break, and the cell will go into cell cycle arrest, it'll apoptose, and problems will happen. So if the problem with telomeres and telomerase is that in a lot of cells in the body, your somatic cells don't express telomerase. Perhaps, it's not clear, most of your somatic stem cells still do. So inheriting something with very short telomeres, the fact that your somatic stem cells can, I think in theory, that's still kind of open to controversy. Open to question, because there was a paper that came out by the Wager's lab from Harvard where they tried to correlate telomerase levels with somatic stem cells in mice and try to use that as a means to track and find somatic stem cells. They showed that for, I think, 1 or 2 cell types, but certainly there's 200 or so different cell types in the body. So, so I guess what I'm getting at is what role there is in, telomeres and telomerase in aging per se. I'm not sure. Certainly having the telomeres too short is a major problem. I don't know if there's a really strong correlation with telomere length per se, that this person's telomere is 10% shorter than that person's telomere. So on average, that person on average will live 1 year shorter. I don't think any kind of correlation like that has been made. So I don't really, I do know that, there's the enzymes that are required to kind of cap or silence the ends of telomeres. So the ends of telomeres also are like, you can kind of imagine them as the plastic that covers the ends of your shoelaces. If you put genes close to telomere ends, they get silenced. There's a lot of condensed chromatin at telomere ends, not just telomerase. So a lot of the enzymes that are required to epigenetically modify other regions also have a role to silence the ends of telomeres. So if the telomere ends shorten, in yeast, if you put in reporter genes, one way to measure the shortened telomere is if you can put in a reporter gene, as the telomere ends shorten, if the promoter is close to the telomere, it'll all of a sudden start expressing that gene. So it'll show you that the telomere is getting short. So having, so it's sort of, I guess, which one's causative, right? So if you have stress, you might also be modifying not just your telomeres, but maybe it's a more direct effect in, enzymes that epigenetically modify a whole bunch of other loci. So, the telomere effect might be secondary. You might also be modifying epigenetically a lot of other things. And as a consequence, your telomere ends shorten. And I don't know, I haven't read any follow-up papers, but I'm kind of vaguely remembering that there was a paper that suggested, and it'd be great if somebody could find this because I don't want to put out misinformation, but that it might be remembering this wrong, that suggested that the telomere ends re-extend or get back to normal once the environment changed or mood altered. So that's, to me, suggests that there might be some effect more on the enzymes that do epigenetic modifications, and as a consequence, not just telomere ends get shorter, but a lot of other things might end up getting epigenetically modified the wrong way because you're lacking an HDAC or you're lacking some other type of modifying enzyme.
Yeah.
Although I'm just speculating at this point.
Cool, thanks. So who's starting an epigenetic sequencing company?
There might be a couple out there already, right?
Yeah, this paper
That will be shut down by the FDA. But that's okay.
Oh, right. Oh, you mean sequencing like 23andMe?
Yeah.
Because I know GeneWiz does that, right?
They do epigenetics?
I think so. I think they do bispecific sequencing. I think they do.
Interesting.
I think there's a couple of companies that do that kind of for service sequencing. So what you could do is you could set up your company as a middleman, right? And you basically provide the interpretation. So you have to just, Yeah, but what we've seen so far from the FDA case is that they're pretty slow. So we'll have like 5 years to really rake it in before we get that cease and desist order.
Yeah, the one of the review paper that Oliver sent actually showed that the current genetic laws don't cover epigenetics. So
Oh yeah, that's
Perfect.
right.
It's the perfect time.
That's a loophole.
Come on. Wait, who's in on this with me?
Wow, yeah, I think you found a golden loophole. Okay, this is on YouTube. Yeah,
23andMe got shut down because they didn't answer the FDA's questions over 40 meetings, not because, necessarily of anything else.
We're 23 and us. Here we go.
Yeah, but all the laws about genetic base DNA, they don't talk about it methylation at all. So there is no legal framework that even touches methylation. So you can do whatever you want from them.
I think we have a resident FDA expert on our panel that can address that question.
No, that the article, what the review article that you sent was saying.
Oh, okay.
And there were like a lot of experts expressing a worry that because it's not regulated, for example, insurance company can misuse it. If they can get their hands on your methylation profile and it matches some methylation profile of psychiatric and stimulus, and they can break up your disability insurance or something like that. So, and people complain that there is no legal framework to protect against that.
Yeah, well, clearly that's a loophole that will probably be closed sometime in the future, but as it stands, I guess that loophole is still wide open, right?
Yes.
All right. Well, this is cool. What are we doing in 2 weeks?
Somebody mentioned, wasn't there somebody suggested, or was it Patrick, the vesicular, some kind of
Yeah, I sent out a paper, like the one, there was one that was just published, and then Patrick sent out a few after that. I can go in 2 weeks. I don't know if I know nothing about this, but I can definitely.
I don't know I still don't know how you mentioned, I still don't know what a startled mouse looks like.
Nah.
We need to write it mail to the author of the article and ask them to send a video.
So yeah, he can send some papers usually do that. They have like attached video of, the maze and all that stuff, but I don't know. Okay, I could do vesicles, some sort of vesicle paper in 2 weeks.
Okay.
I think we should, Julie Wolf's on this list, right?
Yes.
Because her specialty is she looks at vesicle secretion by pathogenic, I think, fungi.
Oh.
So that's what she does, and she was actually designing an assay to look at increased or decreased vesicle secretion. So she's like our vesicle secretion expert.
Yeah, I think
that's cool.
It's a topic we will get her interest.
Yeah.
Maybe she should be leading the discussion then, or at least present.
She's on my list, so
Okay.
She's that stuff going on.
Okay, cool. All right, well, I'll pick one and send out some background info in the coming week.
Cool.
Okay.
Any more epigenetics questions, follow-ups, implications? I mean, you could take these implications really far. I mean, you can, if this holds up, right? I mean, I could imagine writing sci-fi articles, like stories about how whole cultures may have like imprinted memories of like past ancient. I mean, this might be a mechanism for locking in like Jungian archetypes, right?
Video games already got you beat, Oliver. A game called Xenogears has done exactly what you're talking about, down with the Jungian archetypes and everything.
All right, well then we can provide the biochemical background to the Xenogears game.
Side note, that's my favorite game.
Frank Herbert wrote about it in the Dune series too. A lot of the stuff that he writes about in his later books, the Dune series, is about how memories are passed on genetically across generations.
Very good.
Yeah, at some point somewhere I read that another epigenetic candidate is a protein profile of the germ cells, because when the germ cells fused, they carry not only DNA, they carry a lot of proteins at particular concentration. And I saw in a paper at some point that was hinted that initial concentration of protein can actually influence the further development of the organism. So there are other candidates.
I found a I was looking up, trying to find all the different types of review articles on this paper, and I came across some website where somebody was reviewing this paper, and I was trying to get some more detail on the startle response, and there was a message board underneath, and I was scrolling down, which is always the fun part of blogs, right, is the message board. Somebody had posted a picture of Lamarck, and right underneath it, it said, there's going to be a new meme probably. It said, I fucking told you so.
Oh.
Okay.
I guess on that note.
That was awesome. Thank you.
Yeah, good job.
All right, y'all.
Yeah,
Thanks, all.
thanks.
Bye. Have a good night. See you soon.